Hope you all are fine. So, do you know that our earth is home to diverse plants and animals, and all of them have common structural unit that is called cell. That is called as the cell. So yes, in today's session, we are going to study about a specific kind of a cell that is known as eukaryotic cells. So the eukaryotic cells, or cells having membrane-bound cell organelles. So these eukaryotic cells, they have intense or extensive compartmentalization. As you can see here, it is because of the presence of membrane-bound organelles inside the cytoplasm. So the blue colored fluid you see inside the eukaryotic cell is the cytoplasm, but it is highly compartmentalized because of the presence of various membrane-bound cell organelles. So you'll also find that organized nucleus inside the nuclear envelope is also present in between the eukaryotic cell, which is the speciality of the eukaryotic cells. And apart from the nucleus, they have complex locomotory and the cytoskeleton structure. Now, this cytoskeleton structure which is present inside the eukaryotic cell is basically, it has the functions to provide, it provides the shape to the cell. Shape to the cell. Yes? Now, inside the cytoplasm, the genetic material which is present in the eukaryote is organized inside the chromosomes in the form of DNA. So the double helical structure that you find that is which composes the DNA, it is present in the coiled form inside the chromosomes. Now the structure of chromosome is like this. You might have seen the chromosome structure is like this. So the double-stranded DNA is present inside these hands of chromosomes like this. So yes, it is arranged in this kind of form inside the nucleus in the eukaryotic cells. So talking about eukaryotic cells, so we have seen that all eukaryotic cells are not identical. Means, in case of animal cells and plant cells, for example, the set of organelles are quite different from each other. For example, in animal cells, it consists of centrioles other than other membrane-bound organelles, which helps in the cell division inside the animal cell. And it is only present inside the animals but absent in plant cell. And in case of plant cells, it possesses cell wall which is absent in animal cells and only present inside the plant cell. And cell wall provides protection to the cells. Yes? And another organelles which is present inside the plant cell are the plastids, which contains the photosynthetic pigments. Photosynthetic pigments which helps in the process of photosynthesis and through which the plants prepare their own food. But this organelle's plastid is absent inside the animal cell. And another important feature is the large central vacuole. As you can see inside the plant cell, this is the large vacuole which is present especially in the plant cells. And it occupies almost 90% of the area inside the plant cell. And it is the characteristic feature of plant cell only and absent in the animal cell. So in eukaryotic cells, all the membrane-bound organelles are suspended inside the cytoplasm. But it is surrounded by a thin layer which is called as cell membrane or the plasma membrane. And the function of plasma membrane is to give shape to the cell. So later, the studies done by scientists showed that the plasma membrane is composed of lipid and proteins which are arranged in a bilayer form. So this one is the first layer and this one is the second layer, and together they are forming they are called as the bilayer, consisting of both lipids and proteins. Now if we talk about lipids, lipids are arranged in a very peculiar form For example, the head of lipid here, the globular structure, the head of the lipids is hydrophilic. Hydrophilic, it means that this end is the water loving. So that is why the head of lipid is towards the outside. And if we talk about the inner structure of lipid, it is hydrophobic, which means it is water hating. And that is why it is pointing towards inner side. Now, this structure, the cell membrane, also consists of proteins which are of two types. First one is the peripheral protein, this one, and the another one is the integral protein. Now, if you look at the peripheral protein, it is present on the surface of the cell membrane. The proteins that you see on the surface of the cell membrane are the peripheral proteins, and the proteins which you see embedded inside the cell membrane are known as integral proteins. So after the invention of electron microscope, scientists studied the cell membrane in more detail. So in the year 1972, Singer and Nicholson proposed the structure of cell membrane, which is called as the fluid mosaic model. Now, this model stated that the lipid bilayer membrane is present in the fluid form, means it is quasi-fluid in nature, which provides fluidity to proteins to move laterally inside the lipid bilayer membrane. As you can see in this animation, the surface proteins you see moving, that they can easily move laterally inside the cell membrane, is because of the quasi-fluid nature of this membrane. Now, this is the structure of lipid bilayer membrane proposed by Singer and Nicholson. So now the function, if you talk about the function, so the quasi-fluid nature of cell membrane provides or helps in the functions such as cell growth, also formation of intercellular junctions, the function of secretion, and cell division. Now, for these functions to happen, the cell membrane requires flexibility so that these functions can be performed smoothly. Now, apart from these functions, another important function of cell membrane is the transport of molecules across the membrane. And it is facilitated through either osmosis or passive or active transport. Now, if you see osmosis, is the movement of water from its higher concentration to lower concentration, and it does not require any type of energy. Now, if you talk about the passive transport across the cell membrane, it is the movement of molecules or ions from their higher concentration to lower concentration. And in case of active transport, the movement of again molecules and ions, now it is from their lower concentration to higher concentration. Now, in case of active transport, it requires ATP for the movement of these kind of molecules across this cell membrane. So now let us discuss about the cell wall, which is present inside the plants only. So cell wall is the outermost covering present in the plants, and the main function of cell wall is to provide shape and protection to the cells. So, also it provides the cell from mechanical damage and any infection. Now, the cell wall is made up of cellulose, hemicellulose, pectins and proteins. It also consists of 3 layers, which is called as middle lamella, primary cell wall and the plasma membrane. So, we studied about plasma membrane, that it is the lipid bilayer membrane lipid bilayer membrane, and as proposed by Singer and Nicholson, that it is also called as the fluid mosaic model. Fluid mosaic model. Now, the next layer is the primary cell wall, which is the first wall, which is the first layer which forms, and it is capable of growth, but it diminishes when the cell matures. And about the middle lamella. So middle lamella is made up of calcium pectate, which helps in gluing the different neighboring cells together. And this whole three layers, middle lamella, primary cell wall, and the plasma membrane, forms the cell wall. Inside the cytoplasm, there are various membranous organelles which are present. That includes endoplasmic reticulum, Golgi complex, lysosomes, and vacuoles. Now, together, these are known as, or they form, the endomembrane system, and the reason being them called as the endomembrane system because they have or they perform the coordinated functions inside the cytoplasm. So in the endomembrane system, let us first study about the endoplasmic reticulum. So yes, endoplasmic reticulum is basically the network or reticulum of tiny tubular structure inside the cytoplasm. So the network you see here, the various folds, is called as the endoplasmic reticulum, which is near the nucleus. Now, endoplasmic reticulum are of two types. First one is the rough endoplasmic reticulum and second one is the smooth endoplasmic reticulum. Now the endoplasmic reticulum bearing ribosomes on their surface. So here you see ribosomes which is present on the surface of endoplasmic reticulum. So such type of endoplasmic reticulum are called as RER, also known as rough endoplasmic reticulum. Now in the absence of ribosomes, these endoplasmic reticulum appears very smooth, and that is why they are known as smooth endoplasmic reticulum. Now because ribosomes are present on the endoplasmic reticulum here, so we studied that ribosomes are the site for protein synthesis. So that is why RER altogether functions in the protein synthesis, and in case of SER, that is smooth endoplasmic reticulum, it is the site for lipid synthesis. So as we have seen that this surface is very smooth, and we also know that lipids such as fats are also very smooth in their texture. So that is how you will remember that smooth endoplasmic reticulum is the site for lipid synthesis. Now another important functions of ER, that is endoplasmic reticulum, are the synthesis of carbohydrates inside the liver, synthesis of carbohydrates, and apart from this function, the another function of endoplasmic reticulum is detoxification of drugs, detoxification of drugs from our body. So the next organelle in the endomembrane system is the Golgi apparatus, and this is the structure of Golgi apparatus. So this is the flat disc-shaped sac, also called as cisternae, and it is stacked parallelly to each other, and it is also present near the nucleus. Now it has two faces. One is the cis face, another one is the trans face. So the function of cis and trans phase is that it helps in the modification of proteins and lipids. So the proteins and lipids coming from endoplasmic reticulum, that is, the proteins are coming from the RER, that is rough endoplasmic reticulum, and lipids are coming from the SER, that is the smooth endoplasmic reticulum. Now these proteins and lipids come and fuse from the cis phase in the form of vesicles, and throughout the Golgi apparatus they get modified and slowly they move towards the maturing phase, which is also called as the trans phase. Now with this kind of modification of proteins and lipids, they are now transported to the lysosomes. Now these are transported to lysosomes and cell membrane where they are needed. Yes? So yes, from this we know that the function of Golgi apparatus, that it helps in the packaging of material. For example, we have seen that proteins and lipids, and also it is the important site of formation of glycoproteins and glycolipids. So the next organelle in the endomembrane system are the lysosomes. So these lysosomes are membrane-bound vesicular structure, means they are present in the form of vesicles, as you can see here, the structure of lysosomes inside the cytoplasm. Now these are formed by the process of packaging in the Golgi apparatus. Here you see that after the vesicles coming to the maturing phase, that is trans phase, so other than the transportation of proteins and lipids, the lysosomes are also formed through the Golgi apparatus. Now, the isolated lysosomes, so these are the lysosomes, isolated lysosomes we are talking about. Now, these are rich in all types of hydrolytic enzymes, means they facilitate in the breaking down of various carbohydrates, protein, lipids and nucleic acid. And they are most active when the pH is acidic. Now, vacuoles are the membrane-bound space which is present inside the cytoplasm. So here the blue colored big large structure inside the plant cell you see is the vacuole. Now, vacuoles contains water, sap or excretory products which are not useful for plants. Now here the reason vacuoles are present inside the plant cell because they contain excretory products. As we know that plants do not have any specific excretory system, so that is why the vacuoles are present inside the plant cell. Now these vacuoles are bounded by a single membrane which is called as tonoplast. Yes? And in plant cell, the whole vacuole occupies around 90% of the volume of the cell. And this tonoplast, being the outer membrane of the vacuole, it helps in facilitating the transport of number of ions and other materials, that too against the concentration gradient. Now, we know that if the molecules or ions are moving against the concentration gradient, means this kind of transport will require the ATP. Yes, that is the energy. So with this we know that the vacuoles generally contains the water, means consisting of water, they maintain the water balance inside the plant cell, and they also have cell sap which maintains the shape of vacuole, and also the excretory products which are not required by the plants. Now in case of amoeba, the vacuole is present in the form of contractile vacuole, which is important for the processes like osmoregulation and excretion. So yes, in this session, we studied about eukaryotic cells and its components such as cell membrane, cell wall, endomembrane system, in which we studied about endoplasmic reticulum, Golgi apparatus, lysosomes and vacuoles. And in the next portion of the chapter, we will be covering the next topics. Hope you understood everything. Stay tuned for more amazing concepts. Thank you.